Yeah, good morning, students. So, welcome you all for this class, Immunoglobulin diversity or generation of antibody diversity. So, as you people can remember, last class we had studied two important things. One is the clonal selection theory, that is the formation of plasma cells in the bone marrow, and the second one is with respect to the structure of the immunoglobulin chains. So in this class, in continuation with that, I'll be dealing with respect to the generation of antibody diversity. So as we have discussed, immunoglobulin chain is a complicated component or structure which is made up of two different polypeptide chains. One is the heavy chain and the other one is the light chain. And these heavy chain and light chain organizes themselves in many different forms to deal with the different types of antigens which they encounter in their routine activity. So if they have to, and as we know very clearly, the antigens are not just one type. There are many different types, numerous types or infinite, we can say. So every time a new antigen enters into the body, an antibody should be synthesized in such a way that epitope should match the paratope of the antigen. And they interact with them and they will neutralize the antigen. This is how the mechanism works. So for this reason, the immune system is evolved in such a way it can generate infinite type of antibodies. And more importantly, it can store it in the form of memory, in the form of memory cells. Okay. So generally speaking, we know very clearly that these antibodies are produced in 5 different forms. That is, if we people want to remember, if you want to remember easily, we can remember like that: go, man, and eat, dog. Okay, where in go we see IgG, in man we'll see IgM. Okay, IgE, IgA, and IgD. So these are the 5 different types of immunoglobulin molecules which are generally synthesized for many different reasons. And we know very clearly which type of immunoglobulin is synthesized against which type of antigen. Example, IgE is synthesized when there is high level of allergens in the body. In the same way, IgM and IgG is synthesized when they're of bacteria and viruses in the body. So like that, there is a variability in concentration of the presence of these antibodies with respect to the presence of the type of antigens in the body. So to maintain this diversity, the genetics of this immunoglobulin formation is highly complicated and is highly evolved and mechanized with respect to human beings. So how exactly this works? So there are genes which encodes these two type of chains with variable connections, okay, with variable types or variable mechanics. So how it is done? So example, as we know the molecular dogma of biology, there is a gene. From the gene, there is the formation of a mRNA. From the mRNA, there is a formation of a protein, and this protein undergoes modification. And this modified protein then functions with respect to their depository, whether it has to function at the surface as a surface protein, or it has to function as a free molecule in the bloodstream. So this is how generally a protein is formed and it functions. For immunoglobulin, it is slightly different because these immunoglobulins are formed from the germinal cells, okay, germinal cells, or we can say the stem totipotent cells, which are the bone marrow cells. And these cells contain a free germinal DNA, which is not differentiated for a specific type of protein formation, as it is very clearly as it is well defined in any other type of cell. So B cells have this totipotent nature in them. So because of that, their gene, or you can say the primary gene, is called as the germinal DNA, or you can say germline DNA. Okay, so we should not mix developmental biology with respect to the somatic developmental aspects of this DNA. It's two different things, but they correlate with each other to certain extent. So here also we can use this germline DNA will now pick up different types of immunoglobulin clusters and join them to produce an antibody. Okay, and that antibody is formed in many different forms using these three gene clusters, that is variable, a chain for variability, a chain for joining, and there is a chain for diversity, okay, which we call them as segments. And this arrangement is based on the number of chains available for the formation of these globulins. This diversity is augmented, or you can say, so there are two chains which are produced. One is the kappa lambda light chain and the kappa light chain, lambda light chain, two light chains, and one heavy chain which is present as a multi-coding gene, or you can say multi-gene family on different chromosomes, so that the mutations will not affect overall protein formation at once. So there is a high level of conservation, we can say. So since IgG or immunoglobulin are product of immunoglobulins are the product of multi-gene family, different kind of immunoglobulins are produced from different genes. So as you can see very clearly with respect to human beings and with respect to mouse, here the lambda light chain is coded on chromosome 22 and kappa light chain is coded on chromosome 2 and heavy chain is coded on chromosome 14 for human beings, whereas for mouse it is on chromosome 16 and chromosome 6 and chromosome 12. Is it clear? So these chains, as I said, there are variable clusters which can be picked up from them for variability, joining, and constant segments. So okay, so as you can read through here, you can see the kappa and lambda chains are encoded by separate genes on different chromosomes. Each gene is composed of three different types of gene segments, or you can say variable gene segments, which includes joining, constant, variability, joining, and constant. So, and all of this are primarily present in the germline DNA in the B cells. So I'll directly jump to the diagram where you can see here, I'll pick one spotlight. Okay, so as you can see here, there are 30 to 35 different variable gene segments for the variable gene segments, and there are 5 gene segments for joining purpose, which can be picked by the process, and there is one constant gene segment in the germline DNA. And from this germline DNA, the selection takes place. Few of them will be taken from the variable clusters, few of them will be taken from the joining clusters, and one constant is taken from the single constant cluster into the B cell. And from this DNA chain, now the transcription takes place, and then pre-mRNA is formed with one variable chain and few joining segments with this constant gene segment. Okay, further, this pre-mRNA is further processed as we know by exon shuffling. The joining gene segments are few of the joining gene segments which are extra are removed, and only 3 component segments will be left out with a poly, a tail, and a leader sequence. Okay, that is one for variability, one for joining, and one for constant. So, and this now undergoes translation, and once translation is done, this forms a protein, and the protein undergoes further modification, which takes place in the endoplasmic reticulum. Okay, and there is a further modification which also takes place after the formation of protein formation. Okay, so that is called as alternative splicing. There are two splicing which takes place in case of immunoglobulin formation. One is at the pre-mRNA level, and the other one is after maturation of the protein, or you can say after the formation of protein. Further, the amino acids are cleaved in such a way that a different types of protein is produced. So because of that, the variability, the variable protein formed numbers will increase. So that process is called as alternative splicing. Okay, ultimately we should remember that because of this mechanism, a gene segment, that is the variable gene segment, and a joining segment and a light chain is formed in this form. Is this clear? So as we can see here, the light chain is formed. In the same way, it takes place with respect to the heavy chain. So in the heavy chain, there we have variable, diversity, joining, and constant, as it is seen in case of light chain. But here you can see the number. There it was 35. Here it is 51 for variability, 21 for diversity, and 6 for joining, and 8 for constant, where there it was only 1 for constant chain. Okay, so these numbers we have to remember, and we have to point out when we are typing the, or you can say when we are describing the formation of heavy chain and the light chains. Okay, so this is the formation of heavy chain. So where we can say V to Vn, from that the variable chain is picked, and from diversity, the diversity chain is picked, and from the joining, few of the joining chain is picked, and from the 8 constant chain, one or two constant chains will be picked, and from there the the transcription takes place from the germinal DNA to the DNA formation, and from there the rearrangement takes place to form the DNA, and from there the RNA takes place. From this RNA, there is protein which is formed, and that protein will undergo alternative splicing to form the ultimate protein, which will give rise to the formation of heavy chain. So this is the mechanism between the formation of light chain and heavy chain. So we people have to just remember the number which has to be noted with respect to the form, with respect to the heavy chain and with respect to the light chain, and we can easily write the formation of DNA from the DNA to the RNA, RNA from to the translated protein. Okay, so as you can see here, there is a nuclear DNA, and from there again splicing. Of course, it's much confusing. We need not have to worry. Ultimately, you just remember the formation of heavy chain and the formation of light chain as it is described in the last diagram. So next is, what is the reason, how exactly the DNA is working here? How exactly the DNA is working here in this case, in this formation, or how the chromosomes are picked for transcription purpose? Because as we human beings are diploid, there is a possibility of 2 different types of chains. Always there are 2 different types of chains which are available, because we have the diploid condition. So from there, one of them is picked. So how it is picked? Only a single chromosome is used to construct a light or heavy chain. Only a single chromosome is used to construct light chain or heavy chain. So which one will be the best? As we know very clearly how the genes are chosen for transcription purpose. So the first level screening takes place, which cell as better dominative in nature and more sophisticated, that chromosome will be picked. Either it may be belonging to the paternal or it may be belonging to the maternal, doesn't matter. So one of them will be taken from chromosome 2, another one will be taken from chromosome 22, and for heavy chain it will be taken from chromosome 14. Okay, so here you can see how it has picked for chromosome 14. It has been picked for one from the maternal part, and for chromosome light chain, it has been picked from the maternal part. Is it clear? So like this, it is choosing itself and forms the formation, and converts that into the protein. Okay, so this is how, and rest of the alleles will be excluded. So it doesn't mean, as you can notice here, of course, I will not go into the detail of the genetical picking of this because it is the germinal DNA. There is a change in picking this chromosome when and how and when it is required. So at one point of age, it may be picking the maternal chromosome, another point of age, it may be picking the paternal chromosome, doesn't matter. But ultimately, it will pick one or the other chromosome for one particular chain formation. That we have to remember and see how exactly those will be picked at different point of time. Different chains will be formed. For you can see for IgA, one type will be picked, IgG, one type will be picked. So like that, varieties of combination can be taking place. But ultimately, we have to remember each of the chain construct is taken from single chromosome. Okay, that's all. It is, of course, we know the mechanism behind this, how the genes are picked for expression. Same way here also, they are picked for expression. But as the availability for diversity is more, we get more different types of chains. That's it. Okay, so you can see how the exclusion, or you can say we call them as allelic exclusion for the formation of light and heavy chain gene clusters. So the same way here. Okay. Then the other theory which tells how there is a different combination is because of the somatic recombination. So what happens? These cells undergoes, as these cells are getting produced, usually recombination takes place in the meiosis, usually generally in the meiotic cells, but here in the somatic cells also there is recombination. So because of that reason, the maternal chain can jump into the paternal chromosome, or the paternal gene component can jump into the maternal gene component. So you can see the variability that brings into the germline DNA. Okay, so this is one option. The cells are maintaining themselves so that they will have more variability. They can create more variability and more diversity in producing the immunoglobulin, or you can say producing the polypeptide. Okay, so that is one thing we have to remember. The second is DNA rearrangement, or you can say you can also say that is splicing and alternative splicing, as I was discussed before, where the exon shuffling takes place such that they are deleted and rejoined in such a way that a different types of clusters comes together. As I said, there are many number of variability, 35 genes with respect to for diversity in case of light chain, and 51 in case, for variable chain, and 51 in case of heavy chain. So out of that, one can be picked and it can be rejoined with the joining and the constant chain. So like that, the arrangement and rearrangement takes place, and those proteins will be checked against the antigens to see whether that works or not. And if it works, they get the signal in the form of cytokines, and they help the cell to multiply. Okay, so one more is with respect to the class switching. The third way, the first is somatic recombination, second one is germinal shuffling. Okay, and the third is class switching, means, as I said, there are 5 different types of immunoglobulins. So we have to also know very clearly how this change takes place. So if there is type G is getting synthesized for certain duration of time, immediately that has to get shifted into type M, and from type M it has to get shifted into type A. So there should be a change in the leader sequence, there should be a change in the rearrangement of the gene. Ultimately, they make sure that they will produce different types of immunoglobulins. So the class determined by the constant heavy chain region, class switching occurs when B lymphocyte stops synthesizing, as I said, one class of antibody and starts synthesizing the another class of antibody. Okay, class switching involves the expression of the same variable region gene segment, but a different types of constant region. So, and the two important proteins which are involved in this mechanism is RAG1 and RAG2 in case of B cells. So remember these two names, RAG1 and RAG2 in case of B cells. These protein works in changing the class from one type to the other type. Okay, so this is how the next is, this is how it takes place, regulating the variable joining diversity and joining recombination segment. As you can see here, the RAG complex, that is, this is variable and this is joining. So this RAG complex binds and synthesizes proteins, and it introduces an heptameric recombination change, or you can say recombination unit, where the exchange of the segments takes place, or the nicks takes place and separates out. So the single stranded nick is then introduced between the heptameric end of the recombinant signal sequence and the codings, giving rise to the free 3-OH group, 3-OH group that mediates the transesterification reaction. This reaction leads to the formation of DNA hairpin loop coding heads, and this cleaves, okay, this cleaves, hairpin loop cleaves the machinery or mechanism and ultimately results in creating a joint. Okay, so and forming a ultimate VDJ segment and complete protein. So to remember specifically, this RAG complex gives a nick and esterifies it and results and gives a positive cleavage, forming a loop, and this loop will have these three segments. Okay, so next is with respect to the formation, as we will go into the detail of the formation of the B cell. So after understanding the molecular aspects of all this, we can see the expression, how it takes place. So here you can see the pre-B cell. Okay, so this pre-B cell now recombines either through somatic recombination or through RAG1 or RAG2 splicing mechanism, or germinal rearrangement mechanism, or by exon shuffling, or by allelic exclusion. A light chain and a specific heavy chain is picked, okay, for combining VJ and VDJ recombination. So ultimately produces a rearranged gene segment, and this undergoes translation. If at all, if the combination is correct. If it is not correct, if the protein, if the gene which is rearranged is not fitting, or if it is not capable enough, potential enough to fight the antigen, means immediately that cell will undergo a programmed death, or you can say apoptosis. Then comes, if at all, if it is correct, it moves to the formation, it moves to the multiplication, that is result in moves to form the globulins more and more by creating more and more mature B cells and plasma cells. Got it? So this is how exactly the formation of the immunoglobulin takes place. Okay, so one more addition to this is the junctional diversity, that is terminal deoxynucleotidal transferase, TDT. So what they do, they also helps in removing the terminal genes, of course, if at all, if they are added and they have to be rearranged. So we will be knowing very clearly with respect to this terminal deoxynucleotidal transferase with respect to genetic engineering. So that extra chains which are left out are deleted out after the rearrangement. Okay, so this is what we have to remember for linking. The ends are linked by the removed by the terminal deoxynucleotide transferase. Okay, so this is hypersomatic recombination. As I said, exactly how the mutation is being protected and how they are replicating, how they are recombining. So you can remember for this every time. And there is a possibility that somatically there can be an induction of mutation during the somatic recombination, which results in a different mutant cell formation within the individual itself, resulting in a formation of a protein which is usually different from any other individual in the same population. So because of that, we may be able to have individuals who will be highly specialized or highly resistant for a particular type of antigens or antibody. Example, even in case of Ebola or in case of a new virus, we have seen individuals forming different types of immunoglobulins, and from them they will copy the gene sequence and they will try to produce the monoclonal chains for them to produce more and more antibodies of that type. Okay, thank you all. Okay, so just to go through this question, it's a very important question. It can be asked for 15 marks, and of course we may not be able to interact. We may not be able to interact. So whenever you people have, if at all, if you people have any doubt, you can contact me at any point. Okay, thank you all.